a new podcast sponsored by To Enlighten Research. I'm Sara Siqueira. Today we are going to talk about challenges in meta-analysis. I'm glad to invite Amanda Siqueira to join this discussion. Hi, Amanda.
Hi, Sara. It's good to be back.
Bringing a little of history to this conversation, in 1904, Professor Carl Pearson, on his paper Report on Certain Enteric Fever Inoculation Statistics, evaluated the efficacy of serum inoculations to treat enteric fever. Then, he apparently described for the first time in medical research some models to integrate results from different studies. Afterwards, Professor Glass was the first to coin the terminology meta-analysis in his entitled paper Primary, Secondary, and Meta-Analysis of Research, published in 1976. In his words, meta-analysis refers to the analysis of analysis.
Indeed, Sara, the Greek term meta means after, beyond. That's why the word meta-analysis expresses an analysis of analysis. Interesting to know that Emerson described that the term meta-analysis originated from the editors from Aristotle's work known as Andronicus of Rhodes because of Aristotle's natural science paper called Metaphysics. At that time, the term metaphysics was considered as not real physics, since it was written after Aristotle's first work called Physics, which was attributed as the first philosophy. This negative connotation sometimes is attributed for meta-analysis conclusions too. Quoting Eger and colleagues, a meta-analysis is like a sausage. Only God and the butcher know what goes in it, and neither would ever eat any. End quote.
Great observation, Amanda. Like I said, since Pearson's meta-analysis, since that time from now, the non-homogeneity of the studies evaluated was considered a pitfall, and these limitations still persist when meta-analysis is not well performed. Effect size derived from heterogeneous data may not be valid to define recommendations. So, the most accurate analysis should be derived from comparable and similar studies.
Moreover, Sara, the quality of studies is fundamental. A dictum from Lancet reminds us. A meta-analysis is like a bouillabaisse. No matter how much fresh seafood will be added, one rotten fish will make it stink. And how to perform an adequate meta-analysis? That's the challenge assigned to Professor Turner. He needs to provide robust evidence to accomplish the requirements of the Australian Ministry of Health in evaluating if physiotherapy exercise after an elective unilateral total knee arthroplasty can be effective to be adopted as a better management of treatment for osteoarthritis, a chronic disease. According to him, patients who undergo knee arthroplasty may still experience considerable functional impairment post-operatively. The effectiveness of physiotherapy after discharge is an important and valid question. In agreement with Cochrane, meta-analysis is the statistical combination of results from two or more separate studies. And Sara, why Professor Turner chose for a meta-analysis?
Well, he needs to demonstrate the whole of the intervention in a high evidence level for health policies. Meta-analysis provides a summary of the results of an intervention when compared to others, while demonstrating an overall statistical with confidence interval. So it seems to be a good choice. And this strategy seems to have good advantages, don't you think, Amanda?
Yes, Sara. Meta-analysis can bring precision when evaluating several studies together. Important to note that alone, small studies usually are not able to postulate assertive evidence. Also, meta-analysis provides data from questions, not answers, by single studies. And it can show how compatible are the effects of study with a broader population and interventions, permits to investigate the difference in effect estimates. Besides, it can resolve controversies regarding conflicting studies, or it can be able to create new hypotheses.
Let's start a meta-analysis. In the case of Professor Turner, the real gap in the literature in this topic of interest can start the justification to conduct a meta-analysis. And what are the essential steps that Professor Turner needs to follow?
First, he needs to start with the literature review and the formulation of the research question checklist. Professor Turner needs to explain the review's rationale based on existing knowledge. He needs to address the existing knowledge versus uncertainties, the importance and the necessity of the study. He also needs to define, based on the existing knowledge, if the study will be an update study or a replication of previous studies. Moreover, he needs to explain how it is expected that the intervention will work, the effects of the intervention, and he needs to think about assessing multi-component interventions and also about evaluating interventions that target individuals' levels versus the population in order to gain equity.
And to formulate the objectives of his meta-analysis, he can use PICO framework: population, intervention, comparator, and outcome. Another important point is that he needs to think about using a broad versus narrow or lumping versus splitting review. For that purpose, it is important to consider the question's breadth and the available sources, which means the review PICO, the pros and cons. And what is known about mixing apples and oranges, Amanda?
Good point, Sarah. For Professor Glass, one of the meta-analysis creators, comparing apples and oranges is only endeavor worth of true scientists. Comparing apples to apples is trivial. So, broad review with various studies can allow us to investigate the reasons for the distinct effects of some interventions. On the other hand, mixing apples and oranges advises against the incorrect mixing of studies in one comparison when intending to measure an average effect.
One important challenge in meta-analysis is to define which studies are similar to be lumped, which means studies combined in the same analysis, or splitted, which means studies that cannot be in the same analysis. The main resolution for such a challenge is to define the objective of the study and acknowledge these problems as a continuum, where studies with huge parameters variations can be settled as lumped end, and the ones with few parameters variation can be settled as split end. Interesting that heterogeneity comes from differences among individual studies and not to the combination of non-similar realities. Also, heterogeneity can be investigated by lumping, but it is important to take into account the possibility of combined effects which can be difficult to explain. However, when splitting review, higher are the chances of not accomplishing a proper synthesis. Quoting Caldwell and Welton. Inevitably, some degree of aggregation across the PICO elements is required for a meta-analysis to be undertaken. Now, about the test for heterogeneity. When applying Cochrane's Q, it is possible to test heterogeneity, which means that a significant p-value corresponds to significant heterogeneity between studies. Important to note that this test can be less powerful, mainly when evaluating few studies. Another approach is quantifying heterogeneity of the studies by using the variability in the sample estimation by using proper formula. And to assess the quality of the studies, it is possible to use some scoring studies systems.
Wow, it's really important to think about many details before running a meta-analysis. Now, Sarah, how to select the relevant sources?
Well, Professor Turner can search from bibliographic databases, platforms, study registers, repositories, websites, organizations, pharmaceutical companies, expert opinions, reference lists, journals, conferences, and etc. And Amanda, how can he define the eligibility criteria for the studies?
Well, He can use the components described in PICO to specify the eligibility of studies. Also, he can define the characteristics to use to include or not a study, such as time, study design, setting, year of dissemination, language, report status, published, unpublished, conference abstracts. One important point is to be prepared to justify the reasons to exclude some studies.
Excellent, Amanda. And there are some strategies to select existing studies by automation tools, crowdsourcing, or previous known assessments. And it's essential to remember the risk of publication bias, selective non-reporting bias, and one way to handle publication bias is including as many as possible numbers of studies, selecting the ones not published, and also searching in clinical trials registration. And Amanda, how to deal with different assessments of the outcomes?
Good question, Sada. Professor Turner has to handle the different types of variables in the outcomes, the different scales used in the studies, such as the Oxford Knee Score, the American Knee Society Clinical Rating Score, degree of motion, quality of life. The effect size is an estimate with no units, that means the magnitude of the effects which can be used for endpoints assessing the same domain. Besides, to compare different scales, it is possible to apply the standardization of outcomes or to combine scores, such as using z-score.
Great, Amanda. At this point, Professor Turner can try to select as many studies to guarantee generalizability, but selecting studies with good quality, selecting comparable studies, and reducing selection bias. He can also standardize outcome or combine scores for the outcome issues. For the comparisons between studies with small and larger sample size, it is possible to use the weight mean of the effect size. Furthermore, he can evaluate risk of bias in analysis by applying sensitive analysis, subgroup analysis, meta-regression, or adjusting the results from each study. And what is important to take in account when deciding for the meta-analysis models, Amanda? Such as fixed effect model or random effect model.
After overcoming all of these challenges, it is crucial to select the correct statistical model, which is strictly related to the goal of the analysis. So, the model will dictate the meaning of the summary effect and how the effect size differs beyond the studies. The main idea here is to select the model based on the sampling frame and not only based on the results of the heterogeneity test. In addition, as defined by Ronald Coase, if you torture the data long enough, it will confess. So, in the end, all data will be tortured. The main question should be, what is this meta-analysis can really confess?
Yeah, Amanda, a supposed scientific discovery has no merit if it cannot be well explained. And we know that not all meta-analysis results are proved to be valid. You still need to be aware of the risks of changing long-standing practices according to one unique meta-analysis. That's why it's fundamental to perform meta-analysis according to standard recommendations to avoid misusing it.
Wonderful, Sarah. And I would like to end saying, the use of meta-analysis in daily practice also requires to see the horizon of its replication, its quality, and its being true, the unconcealment of science.
I highly agree, Amanda. Thank you so much for being with us today.
My pleasure, Sarah.
And I hope to see you in the next podcast. Bye bye.